So good afternoon, everybody. My name is Jesse, and I am your virtual adventure guide here with Exploring by the Seat of Your Pants. I know we've got some familiar faces in the crowd today, but if you are a student new to us, we are all about bringing conservation, adventure, and science into classrooms around the world through over 40 live, free monthly interactive broadcasts. Now, every single day, we get the chance to hang out with some of the coolest scientists and explorers in the world, but today's program, even by that standard, is pretty special. Last week, we got the chance to hang out with the team at Sea Women Expeditions on their ship off the coast of Norway, doing some really cool science and exploration and sharing their passion and enthusiasm with kids like you. We got the chance to hear from Whitley and Sarah talking about orcas, and today we're diving in with one of my very favorite kinds of science in the world, and that is eDNA, or environmental DNA. So I'm going to bring up the link if you guys want to learn more about Sea Women Expeditions. I'll make sure everybody has that for the end of the broadcast. If you want to check out our Orca Talk 2 from last week, it is on our YouTube channel like everything else. But right now, we're going to dive in with Expedition Leader Susan Eden to introduce our two main speakers for the day before we dive in. So Susan, Alicia, Wing, thank you so much for joining us, and take us away, Susan.
Good evening, everyone. It's 7 o'clock here in the Arctic in Norway, and we're really excited to talk to you all. We've got a little bit of a time delay, but my name's Susan Eden, and I'm the expedition leader of Sea Women Expeditions, and we've been in Norway for 3 weeks on the ocean. This is cohort 3. We spoke with cohort 2 last week. I'm from Canada. I live in Yellowknife in the Northwest Territories in the subarctic, and I'm a geologist and a geophysicist by profession. So we're going to talk about eDNA and marine biology, which is really different from geology and geophysics. They're all interrelated to a certain degree, but I've been getting to do some of the eDNA work as well, which is really exciting. I want to introduce our two lead scientists on cohort 3. To my left here is Wing, Dr. Wing Yan Chan, and she is a postdoctoral fellow at the University of Melbourne. And she's originally from Hong Kong, and Dr. Yan Wing Chan is an expert in marine science. On my other side is Dr. Alicia Bruzos, who's originally from Spain, and Alicia is doing her postdoctoral fellowship at the University College of London in England. So we've got two really exciting scientists here. I want to turn the floor over to them, but I just want to thank them for running the science program on this cohort. It's gone extremely well, and I hope that we've got great data that comes out of this program, and we'll know what we've got in terms of eDNA, or animals living in the ocean, probably in a few months once the data is all sequenced at the University of Waterloo in Ontario. Take it away, Alicia and Wing.
Thank you, Susan.
Thank you, Susan. So we have the official introduction about ourselves, but now we have the unofficial introduction. We're going to tell you a fun fact about Alicia.
Okay.
Yeah.
What do we want to share with them?
Alicia, we have a swinging competition a couple of days ago, trying to see who can get to the highest. Well, Alicia is just an expert that there's no way I can match with her, so she can swing really high.
But don't worry. Here you have Wing, and you will never forget her name, because she's able to run the machines that fly in the sky. So she's a pilot of planes and helicopters. That is awesome, actually.
Oh, thank you.
And you're able to stay in the sky and also in the sea in this expedition, right?
I can't catch up with you on the swing, so I just need to take off and fly. That's
maybe we can teach each other something.
I think so.
Yeah. Well, let's move to the eDNA, right? We don't have much time.
So to understand an environment and to protect the environment, there are several information that we really need to know. So important things are, what are the species, where are they located, and how many of them we can find. So let's imagine in this beautiful marine ecosystem here, this is a tropical coral reef. If we were to understand this environment, how are we going to do this, Alicia? How do you think we can find and understand the species and the numbers?
Well, we can actually count, right? If I stand here, I will count 1, 2, 3, 4, 5, 6. Although, yeah, maybe at some point I will mess or forget the number.
Yeah, so counting is a common way we use in science to identify how many species, how many of them, and where they are. But there are problems associated with that.
Are they?
Yeah.
Which ones?
For example, let's have a look at here. Can you see a fish?
Oh, you were right. There is something
And
there.
a cling-on fish
I
here.
didn't look at that at
Yes.
the beginning.
So these species, these are called cryptic species. So they are very shy and they're hiding. They are really hard for us to visually see them.
You're right.
Now the problems are these two fishes. Do you think they are the same species?
Yes, they look very alike,
They
right?
look very alike, but that doesn't mean that they are the same species. Sometimes
So-
they can be different.
Are you telling me that those are not the same species?
Yeah, they are not the
Okay.
same species. Even though they look the same,
Interesting.
they are genetically different.
Hmm.
The other bias that we have is we always like to see the big thing.
Yeah, no, definitely. That is the first fish that I spotted when you put this picture in the screen. So yeah, you might be right.
So when we're counting it, we're looking for the big fish, the really bright yellow, colorful one. They are the one that count first. While the little one and those that are not colorful, we tend to miss that. So we're missing a lot of data that are very important for conservation because we don't truly understand the environment if we don't have organs of them.
Yeah, what a pity. We really
Yeah.
like to understand the environment, right?
So how do we fix this?
Well, let me think. Imagine that you want to understand the cake of your last birthday. So which cake do you usually like?
I love chocolate cake.
Chocolate cake.
Yes.
Okay. Let's see how can we know if her birthday cake had chocolate on it. So what could you do is to go and find a book of recipes, and then you will pass the pages, and oh, voila, there it is, the recipe. And you can read it and find out if there is chocolate or not in that recipe, right? So, do you remember that thing that you studied in the biology class that is called cell?
I really can't remember. Please tell me.
Well, all living beings, we are made of thousands and thousands of cells. And sometimes we lose them, I must confess. So,
Our hair, skin, everywhere.
yeah, everything is a cell in a living being. So, in the cell, in the middle of the cell, in the nucleus, we found this molecule that you are seeing now in the screen, so the DNA. And the DNA is like the recipe. It contains all the information. So it will say if you have the straight hair or curly hair. It will say blue eyes or brown eyes. It will say tall or short.
Not that short.
Very short. So everything is written in the DNA. So if we are able to read the DNA that is in the fish or in the environment, we will be able to know what it is in there, right?
That sounds really exciting, but how do we read the DNA? How do we see it? How do we read it?
Well, it's very similar to the recipe if you think about it. So how do you read the recipe?
The recipe has got letters in it, so I read chocolate, cream.
Yeah. Okay, so you use the alphabet and you read the chocolate part,
Oh, you're
right?
right. Yes.
We can do the same with the DNA. So the DNA, it's written only with 4 letters. So not 3, not 5, 4 letters. Everything is written with only 4 letters that you can see on the screen, A, C, T, and G. But don't worry, if we organize them in different order, the words that will come out of them will be different. So a crab, for instance, will have the CTGHG, right? However,
What about fish?
the fish will be different.
Oh,
Can you see the difference?
Yeah.
And every living being has DNA, so even the algae will have the DNA. Can you see? It is also different.
Yeah, and this is how we can then use it to understand the environment. So eDNA stands for environmental DNA. When we collect this DNA from the environment and then sequence them, it would then tell us these different marine species that we can identify.
So let's now dive a little bit into the field and see how can we collect this eDNA. So in the environment, there are animals and they lose cells, like you when you're sleeping, for instance.
They also poop.
Yeah, that also contains DNA actually. And those cells that are released to the environment will have also the DNA.
And once we collected this sample and we're able to sequence them, and we'll be able to find out what this DNA is associated with. It could be a fish, it could be a crab, it could be an algae,
Or even a frog.
it can be anything. Could
Yeah.
be you.
Yeah,
I think Olivia.
could be me. If you go to my bed, probably you will find the skin cells in the bed.
So this is a cartoon though, so how do we do it in real life?
Yeah, so let's put away the slides and have a dive into the reality.
And we will show you how we've been doing sampling at the Sea Woman expeditions.
Yes.
Okay.
So as you can see, we are here in the boat. And the first thing that we have to do to go outside,
Safety first.
safety. So will it be cooling?
We're pretty cold out
cold?
there. We're in the Arctic Norway, so we must have some protection.
Okay, I will dress myself then
Yes,
with this awesome jacket.
let me help you.
Thank you, Wing.
So now she is protected, nice and warm. She is also highly visible because we're sampling in the dark. We need to find her in the dark. We also are near water, so we need a life jacket as well.
Life jacket.
Wonderful.
So now I'm protected. I can go to the sea, to the Arctic Sea.
Fantastic.
Perfect.
Now, so we are ready to go, and then we can go for sampling.
Okay, perfect. Let me see if I have something.
What have we got?
Look at this. Look what I found. What an
Oh.
awesome machine.
This is a pretty good gadgets that we use to collect seawater sample to identify the eDNA. How does that work, Olivia? Can you show me?
Well, you will have to open the tube in order to collect water inside the tube. Could you do it?
Yes. So we want to collect water from a particular depth, and we can't really simply use a bucket and hold it up, because as you hold up that bucket, you will be collecting seawater all along. We want to be a particular depth, and it stays there. So we have this specially designed machine that can do the job. So how it does is we can open this. Okay, lock it.
Are you ready?
Yeah, I'm ready.
Perfect.
Right, so now this is being opened, and we can deploy that into the seawater. Once this,
Yeah, one is down there with water inside. How can we close the doors? Are we going to dive?
it's going to be a little bit cold for going for a dive, so we have something a little bit more clever than that.
Okay, so
Here we go. Olivia has got this.
maybe we can
weight
use
here.
this weight and launch it from the surface,
Yeah,
and this will knock here.
so we
Release
say nice
this.
and dry, and then Olivia can then release this.
Yes.
Here we go. Action. Wow. Okay.
You see? The door's closed.
So now the seawater sample is being contained within the sampler, and then it would not be contaminated as we hold it up. So we can have the specific DNA collected from a particular water depth.
Perfect. We have the DNA. Do we need anything else?
Yeah, we do want something else as well. For example, to understand this data, we want to understand how much visibility are there in the seawater. Because if there's not much visibility, that means the water is not clear. There can be a lot of little organism living there, or there may be no organism, not much organism living there. So the water is very, very clear. And there are ways that we can measure that.
Okay, let me see. Let me see if I can find something.
I'll show you.
Oh yeah, I could.
Here we are.
Look at this. A seki disc.
That's how it works.
So can you see black and white? It's a very easy thing to see, right? So it says that.
Okay.
So we can drop it. I will drop it. And we are going to put it very deep into the ocean and keep checking if we can see it. Let's try.
So when we are measuring for visibility, we keep deploying that, let it go down to the ocean. At some stage, you won't be able to see the color anymore.
Can you see it, Win?
I can still see it.
We have a good visibility today,
Very
right?
good.
Nice.
Come back. We are obviously on dry land at the moment, and we will show you an actual video of how it works. So whenever we are ready, so this is us collecting sample. We're collecting sample in the morning and afternoon twice a day. So here we are deploying this secubis. So you can see the black and the white color now, and we are slowly lowering that into the water. You can see it very clearly now, but as we further go lower and lower into the water, it will start to disappear.
Just like here? Or will
Let's
it
have a look. We can
disappear
still see
earlier?
it. Can you still see it?
Yeah, I can see it.
Yeah, the white color is very
I can see it.
easy. You
I can see it. I can see
It's
it.
starting just to disappear. It's not as
Yeah, but I
clear
can
now.
see it.
Oh, yeah, yes, you're right. We can
Yeah.
still see it.
Yeah,
Still there. It's starting just to disappear.
almost there.
Almost there.
Can you see it now?
I can.
Oh,
Just. That means it's at the depth that the water is not so clear anymore.
I cannot see it anymore.
I think it has disappeared now.
Yeah.
So this is the time that we will say, this is the water depth that we have.
Okay,
This
the
is
visibility.
the visibility, yes.
Yes.
And we'll let the team upstairs to know this is me calling out 10 meters.
And I will note in our notebook, right? Because we like to take notes.
Exactly. So after knowing this, we now want to show you how do we do water sampling.
Oh, that is the machine that we were playing with before, right?
Yes.
Nice.
Let's look at it in action in real life. So here we are, slowly
Oh,
deploying
the doors
water.
are open.
The
like-
doors are open, yes, because we need that to let the seawater come in to the instrument.
Oh, yeah, you were right. It was for that.
So now it's coming down. We're counting how many meters. We have a little marker on the string so that we know exactly how many meters is it going down to. And we consistently collect the sample at a certain depth.
We cannot see it anymore.
We can just see, but this is not the second dish, so it doesn't matter, we can't see it.
Yeah, you're right.
So this is coming down to 6 meters for the sampling. And now you can see
Wow.
that big splash there, that is the weight coming down. So now it will boom, activate the door, and now we close the seawater sample.
And you will only have the water that was down there, not the one that you are
At that level
coming up.
only.
Interesting, a smart way to take samples from the deep ocean.
Yeah, so now it's coming up. As it's coming up to the water surface, you'll be able to see that the door, the two blue doors, are now closed.
What if they are not closed?
They're not closed, then we have to do it again.
Again?
No,
But that might be very cold.
that's okay.
How many times do we have to repeat it?
Most of the time we can get it done in 2 runs.
Okay.
Sometimes we can't close the door because there was strong current, so the device really going away. The weight can really hit the door properly.
Okay, now I understand.
So now that this is ready, we'll take it out and then put the seawater into the bottle. This bottle will then go into our laboratory inside, and then we'll do some filtering.
Okay, that's interesting. And that water, what it is for?
This is for the eDNA that we were talking about. So the seawater now contains all the leftover cells, skin, hair from different marine animals.
Oh, like the video that we have seen, that with the fish pooping
Yeah.
and the animals releasing hairs?
Exactly.
Okay, now I understand.
So from the seawater, we can detect small things like phytoplankton. So there are little plants that's swimming in the ocean. We can detect also big things like orchids, humpbacks as well.
Wow.
And from using this sample, we can then build the environment. What's around here? What kind of marine species can we detect? And answering the question of species distribution and abundance, which are very important for environmental conservation.
Yes.
That is particularly important in the Arctic Norway that we are here, because the Arctic is one of the regions that experience the greatest change in climate. So the area is warming up, and therefore it's very important for us to understand this marine environment. This will help us to link what happened in the past and in the future, and then we can collect data to inform policy and conservation. And speaking about policy and conservation, Sea Women Expedition is really just not a team of scientists. We have storytellers, we have artists, lawyers, a lot of different expertise on the team. I'd like you to introduce Leslie. Leslie is an environmental lawyer from Toronto.
Thank you for introducing me.
She's been helping us with water sampling in the last couple of days. And tell us a little bit more about you.
Sure. I'm an environmental lawyer in Toronto. Like we said, I also practice Indigenous law, including I'm grateful to be able to practice as a lawyer for my own community, the Bonnesh Algonquin First Nation. And I've been a scuba diver since I was about 12 years old. And so I am loving getting to be here and assisting in research that helps protect our oceans.
So from all the machines that we have been looking at, the instruments, which is your favorite?
I think the Sucky disc, because I love that I don't need to have a ton of science experience to be able to help and make a big contribution, because I can just stand on deck from a different vantage point and tell you when we can no longer see it. And that's how we get those data points.
So you want to be a scientist now?
Maybe.
Okay.
What does it mean for you as an environmental lawyer to be involved in a science project?
I think it's really important as an environmental lawyer that I get this kind of fieldwork and science experience, because this is the kind of science that my clients rely on through environmental assessments or through challenging decisions in court. And better understanding this research and how it's done helps me better serve my clients and come to better decisions to protect our environments.
Amazing. And that really speaks to the importance of the Sea Women Expeditions. We are not just a team of scientists. We have artists, storytellers that can really help to present the story of the research to different people. We have people like Leslie and they're environmental lawyers. And once we understand the data, she are the type of people that can help us to bring it to policy and conservation. So together as a team, we need all these different expertise to make a difference to protect our environment for the future generations.
Yeah.
Yes. This is our eDNA project.
Oh, Leslie Ann Wing, Alicia, this has been so much fun. You put so much care into creating that presentation. It was extraordinary. So thank you so much for that. And we're going to dive in with questions. Our YouTube groups, if you want to share in the chat bar there, we'd love to hear from you. Miss Hurley, Miss Denica's class, we'll come to you guys in just a second. You can let those questions percolate for a minute. But we're going to dive in with one that we got written in from one of our classes in advance. And so they want to know how many species you've been finding. So when you've been doing these analyses off the boat, how many different kinds of creature are you actually finding, or things in the water? Or do you have that data yet?
So we don't have the data because we are collecting the water. We cannot have the data unless we sequenced. And we have to do that in a molecular biology lab. So a lab with lab coats and with the strange liquids and pipettes.
Goggles,
amazing things.
Gear.
But yeah, we cannot do that in the boat because it requires a lot of big machines and instruments. So here what we are doing is dressing ourselves, right? Every morning, very early, too much.
And every afternoon.
And every afternoon, yeah, you're right. You remember better than me. You should be the scientist. And we collect the water and we conserve it in a cold.
We keep them cold, so it's we're able to sequence them when we send that to a molecular lab.
Yeah. So unfortunately, we cannot answer this question now, but maybe if you ask us in 2 months or so, we will be able to tell you something.
We're going to come back then and ask it again just for fun. Miss Hurley's class, I'm going to head to you guys now in London. Welcome in Ontario. If you want to come in and ask us a question, you're good to go. Hey.
Hello. What animal in the ocean has DNA that is closest to human DNA?
Ooh. Did you
So human DNA, we're mammals, right?
Yes.
So maybe the whales? Could they be the closest to the humans?
In the marine life that is closest to human, marine mammal would definitely be one of the closest to us.
Yeah, I think so.
So orcas, humpback, that we actually have seen them today in action in water. So we saw orcas while we were snorkeling.
Cool.
So yeah, probably marine mammals.
Yeah.
Yeah.
So for our students, the other marine mammals, we've got all the whales, dolphins, porpoises, we've got sea otters, we've got dugongs and manatees. There are a few different groups of mammals that have made their way back to the ocean. So that would be the closest. I don't know if any of those are the most related to people of all those groups offhand, but I'm really glad we got that question. Great question, guys. RFIS Intermediate, Miss Enica's class, Flemington, if you guys want to come in and ask a question, you're good to go. Hi.
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 You have to ask a question.
Yeah, ask away, man.
How long can polar bears survive in water?
Ooh, if you ran into a polar bear, how long is it stay in the water?
Polar bear. I am not a polar bear expert, so I'm not familiar. Does anyone know how long they can live?
Yeah, please
Go,
come,
Susan.
join us, Susan.
Everybody come in. Hey.
Hi. Can you see me?
Yes.
All right. You know, in Canada, polar bears can swim a couple 100 kilometers at one time. So they've got really thick fur, and they're designed to swim long distances when they're hunting and when they're going to land after the sea ice has melted for the summer and they need to go to shore. So they can survive a very long time, probably a couple days at least, swimming long distances from one island to the next, or maybe an island to the mainland. So they are equipped to swim really long distances in the Arctic, and no other animal probably can quite swim the way a polar bear swims. So that's the answer.
I love this question. We've actually got a couple polar bear programs coming up next week if anyone's keen, but their Latin name is sea bear. Like that's what it stands for, because they're classified as a marine mammal. They hunt on the ice, they hunt things in the ice. So they're a really special creature that sort of exists at the interplay of the two worlds, land and sea. I love that we can just keep bringing in different experts. Like we want to fill the room. If there's 50 people on the boat, they should all end up in the broadcast by the end, I think. I'm going to take a quick question written in for us, and then we're going to head back to Miss Hurley's class live. So once you get back to the lab, you have these data tubes full of the water. Can you tell abundance? So you can tell that there's a certain species out there, but can you tell, oh, the ocean's teeming with this kind of fish, or there's just one out there? Is there a way to identify that from the data you collect?
Yes, so the sequencing method we use is called metabolic coding. So what it does will give you what we call relative abundance. So within your sample, it will tell you the proportion. Let's say 20% of them belongs to a particular phytoplankton, maybe 10% belongs to a jellyfish, 30% of a herring, 10% of the humpback whale. That would give you the community structure in percentage.
Yeah. Very cool. I love it. Miss Hurley's class, you're right at the camera again. Come on back in. Hey.
Hi. What was the smallest cell you have ever seen?
The smallest cell?
Yeah.
Yeah.
The smallest cell would be a virus, right? Which is not a cell.
It's not a cell.
No.
Do you mean a marine organism, or do you mean any cell in general?
I think any cell,
Like
when you're doing this research, is there like creatures or organisms of any kind with really tiny? I like that you mentioned viruses. They're a little bit different than other living things, but do certain organisms have smaller cells than other ones or not?
I work with some very small marine planktons, not plankton, marine microalgae. So a particular species I work with is Bedonoflatula. So they are between 5 to 11 microns. So they're very, very tiny. That Olivia is a cancer researcher.
Yeah.
So she actually worked at the cellular level. That's a different story, you can tell.
So yeah, I work with cancer when I'm not here in the expedition. And cancer cells in general, at least the cancers that I'm studying, they are very big cells compared to others because they have a lot of DNA inside the nucleus. So this thing that we have been seeing to read letters, the cancer cells, they have a long book, a very big encyclopedia inside the nucleus. So they are big cells actually. So that won't be the smallest cell, unfortunately. But yeah, I don't know. We will have to research a little bit about the smallest cells. We have something to do after, right?
Yeah, I always love questions where we sort of stump the scientists, but those are some great answers. And what a small thing, 5 to 10 microns across is crazy. I'm going to put up a video in our chat for anyone who's keen on cancer cell development too. There's some really great stuff on YouTube about that. But let's head back to RFIS International. If you guys, or Intermediate, if you have another question for us, come on in. Hey.
I was wondering, the machine, when you're lowering it down into the water, how do you keep the water from flowing into it when the doors are open?
Yeah.
That's a very good question. So as you lower that very cool water sampling device into water, the water will keep coming into it. But because it will come stop at a certain level, so the water is being exchanged constantly. So let's say it is coming down now and it's open. Alicia, please help me. This is open. This tube will now exchange the seawater with each step. So if we're going down further, it will now collect seawater from this step. So we don't have to close it on the way down, but we need to close it on the way up.
Okay.
We close it when it is down, and then we pull up, and no water is entering.
So water's flowing through the whole time you get to the depth that you want. Now, out of curiosity, how many different samples do you take? Like how many of these tubes do you have, or filled up sort of data samples do you have on the expedition so far? Is it 10? Is it 100? Is it how many?
So we do 2 every time we go. We do one at 6 meters and another one at 8 meters. And we go twice a day. So that will mean 4 samples per day. How many days have we been sampling?
We'll be sampling 20 days.
20 days.
In total.
So 20 days by 4. Please, mathematician.
80.
Perfect.
Hey!
Yes, she's still doing the maths.
Why those depths? Like why choose those specific depths? Is there a reason that maybe the water column is different in those places, or you know that different creatures live in those areas? Why them?
That's a very good question. We don't want to go too deep because one of the particular marine species we are interested in are phytoplankton. Phytoplanktons are plants, so they photosynthesize. If we go too deep, there's no light, and they can't photosynthesize. There won't be too many of them. So we want them to be a little bit higher. When it's up in the surface, it's difficult to be very accurate because there's a lot of surface mixing in the surface as well. So we've chosen a depth that is 6 to 8 meters that will still help to answer the question of photosynthesizing planktons, but it won't have the problem of the surface disturbance. That's what I think.
Yeah, and that's why we use the SECIDs to see how far the light can go. So how is the visibility? How far can we see? Because that is how much light this phytoplankton is going to have.
Yes, and we got more data than that as well. So we have eDNA, we have visibility. We also have another device over there. Alicia, you can help.
Yeah, we can show more
We got more
amazing
toys.
tools.
So this is the
I'm dropping
CTD.
everything.
So this is conductivity, temperature, and depth. So what it does on a daily basis, when we are sampling the water, we also lower that into the water column. That go all the way down to 20 meters, and then we'll pull it up. This device, as it's going down to the water, it would tell us, it would continue to log the temperature, the salinity, conductivity, and also the depth. So we're able to bring a profile of where from our sample, where they come from, and then surrounding environment around it. So the temperature, the salinity, for example.
Spectacular.
And what about this one?
Go ahead.
This is a GPS.
Yeah, what does it give us?
That is very important because as we are sampling, we need to know where are we. So this Garmin GPS will then tell us our latitude and longitude. We can then link this data point is where we collected our data. This is also very important for other scientists coming to do the research. They will know at this point we have collected the sample, and this is the marine community that we can see, and they can relate it to future data that can be relevant to understanding climate change.
Yeah.
Spectacular. And I also, way to go talking over the broadcast system on the ship. This is the challenge of doing a program when you're live on a research vessel. Very, very cool. So thank you for that.
Sorry.
No, you did great.
Channel. It's actually the Marine Channel, Jesse, of the Norwegian Coast Guard giving the update of the weather, because we're on a vessel and we're in the ship's bridge right now where the captain steers the boat. So that was the Marine Channel.
That's about as exciting as it gets. I wish we could switch positions, but I'm not allowed to come. It's Sea Women Expeditions. We are featuring some really incredible women from around the globe. So I'm so happy. Anyway, it makes it so much better in the broadcast when something like that happens. So thank you. We're going to take a couple more questions. Time flies and you're having fun. So we're nearing the end of the broadcast. But Miss Herbie's class, you guys have a student right up. Come on in. Hey.
Hello. How much of the ocean have we discovered?
Ooh, that's a difficult one, right?
That's a difficult question. And interestingly, we know more about the space than the deep sea. So,
but we know more about the Earth, the terrestrial environment, than the sea.
Yes.
So we are not that bad.
Ocean is really big, so our knowledge on different level of the ocean are really different. We understand a lot on the surface level. So the phytoplankton that live there, the coral reef, the big species, orcas, humpback, because they're easy to see and they're easy to reach. However, we know very, very little about the deep sea. Deep sea referring to the depth of 5 kilometers down to the ocean floor. We only have explored a very, very small proportion of the deep sea, and we indeed know more about the moon than the deep sea at the moment.
So if you want to be biologist in the future, which is an awesome profession, maybe you should focus on the sea instead of the earth, the terrestrial environments, because there are not many things that will surprise us probably, while in the sea there are so many things that we don't understand and we don't know how they work, that you will be doing eureka all the time. Eureka, I found this. Eureka, I found that. So yeah,
there are still things that we don't understand on land, on the terrestrial environments. For example, new insect species, bacteria that we still don't know. But comparatively, the deep sea is so little that we know about it.
We have on regularly an amazing speaker named Diva Amon. She's a deep sea explorer, one of my favorite scientists we've ever had on the broadcast. If you want to check out her programs, they're on our YouTube channel. But she talks about how we've had scientists on who have come on and we've asked them, how many species have you found? How many new species? And one of them said, I don't even know. Like every time she explores the deep sea, she finds that many new species that she doesn't even have in her mind, how many new things to science. And that's what's so exciting about biology. I mean, you two have done such a beautiful job of sharing your passion and being all excited about this today. But every time we have on scientists who are ocean explorers, it's such an incredible realm. It's 70% of the planet. And you get the opportunity to work with people from around the globe, do incredible things, be on the deck of a ship for a big announcement to happen, and just have a ton of fun. I mean, if there's a better job than that in the world, I don't know what it is. So I'm so glad we got the chance you shared all that. Miss Denica's class, New Jersey, we're going to head back to you one more time for one final question before we keep the learning going and share how you guys can keep following the expedition. But RFIS, come on back in. Hey.
It's Charlotte again.
Hey, Charlotte.
I was wondering about how many organisms and like DNA strands do you find in one sample?
Yeah.
We can find many, because cells are very, very tiny. We were talking about this before. So we are taking 1 liter each time we sample. So in 1 liter, there can be many cells, and they can come from different individuals. And yeah, most of the times they won't be cells, they will be just DNA floating, and all of that, those cells and that DNA will get sequenced with this metabarcoding method that we was talking about before. And once we have that, we will have lots and lots of words, like the ones that we have seen in the presentation of fish, crab, whatever. And all those words need to be analyzed. So we need to sit in front of the computer, do like a lot of coding, and then we will be able to see how many do we have in each 1 liter sample that we are taking. But yeah, for sure we will have plenty of them, right?
And how many organisms, really depending on where you sample. So you maybe happen to be sampling near a herring run, so you might find a lot of fish DNA on that. You might be in somewhere that is quite barren, so there's not much over there as well. So therefore, it's quite very important for us to figure out where we are. So we have the latitude, longitude, and it helps us to link to the data of what's around the environment.
Very, very cool. I'm so glad you mentioned a herring run too. I encourage all our classes to check out herring runs. They're one of the coolest things in the whole world ever. And I also like that Alicia does computer stuff like I do, just 2 fingers really rapidly. That's how I do stuff when I type around as well. Yeah. Alicia, Wing, Susan, Leslie Ann, it's been such a pleasure having you with us today. This has been a beautiful program, a great follow-up to our orca presentation, and such fun hanging out with you all at Sea Women Expeditions. Again, for our classes, if you want to check out the website, all your social media pages, they are below. Is there any last message you want to share with our kids today about the work that you do, about eDNA, before we wrap up and bring them in to say thank you and goodbye? Any final parting words?
I would say to any girl that is in the class that you can do it as much as any boy. So just come and join us. I mean, women can do science and it's really fun.
Definitely. Hashtag science cool.
Yeah.
Like, on both of these notes, I will say we have about, I'd say about 65 to 70% of the speakers that we bring on are amazing women from around the globe. Soon we've got February, which is our entire month only dedicated to awesome women in STEM. So when I was in university, it was like me and a sea of women in the biology department. It was spectacular. So it's incredible the work that's being done on Sea Women Expeditions. There's so many other ways to learn about girls in STEM. If you're keen, I really encourage our class. Actually, for all you, while you're here as speakers, after our first presentation, we had classes follow up to say, hey, like, send us more about this expedition, send us more about amazing girls in STEM. We'd love to hear from you. So I think it's just a spectacular expedition you guys are on. Thank you so much for joining us today. And I hope kids take the opportunity to learn more. What we do now, as you know, Susan, because you were on the other broadcast, I'm going to bring in Miss Hurley and Miss Denica's class to say a big thank you and farewell. Miss Hurley, Miss Denica, thank you so much for joining us.